Okay. Today our speaker is a visiting scientist from China Agricultural University in Beijing, China, where he is an associate professor in the College of Animal Science and Technology. He earned a Bachelor of Science in Public Health and his PhD in Medical Molecular Immunology from Beijing University Health Science Center's Department of Public Health. He's published in many journals, including Journal of Nutrition, Clinical Cancer Research, Journal of Animal Science, Frontiers in Biosciences, Journal of Agricultural Food Chemistry, and Food Chemistry and Biochemistry journals. He serves as a reviewer for more than 16 journals, maintains memberships with the American Society of Nutrition, the American Chemical Society, and American Society of Animal Science, as well as serving as a council member of the Chinese Swine Industry Technology Innovation Alliance. And he's also received several awards for research and teaching. In 2009, he was awarded first prize for technology progress of Jilin Province in China. In 4 straight years, he was awarded the China Agricultural University's Excellent Teacher and Faculty for the National College Students Innovative Experiment Plan. And then in 2014, he also won the Young Scholar Award. His research interests include amino acids and lipidogenesis and myogenesis, efficient utilization of dairy nutrients, and intestinal health, and soybean anti-nutritional factors. While here at Texas A&M University, he has been studying intestinal amino acids and bioactive peptides with Dr. Guoyao Wu. So please help me welcoming Dr. Ji Ma.
Just take a moment to remind you, graduate students are invited to have lunch with the speaker in the faculty lounge immediately following the seminar. So some of you think off so fast, you may miss that opportunity. Sorry to interrupt.
Thank you for the introduction. Thank you. Many thanks to Professor Chapin and Professor Guoyao Wu giving me this chance to have a talk with everyone. And it's my great honor to have the chance to give my presentation about my work in China, focus on the adipogenesis and meat quality. The title of my presentation is Modulation of Novel Gene CREBL2 and LI2 involved in adipogenesis. I am from China A&M University. Now I am waiting scholar in Dr. Guoyao Wu's lab. As we know, with the development of the abundance industry, the intensive genetic selection of animals with lean meat rows have significantly reduced the intramuscular fat and impaired the eating quality of meat. And so the intramuscular fat is a crucial factors to influence the meat quality. How many factors we want to ask? How many factors have influenced the meat quality? The answer is many factors, including pH value, drip percent, and marbling, and the length of meat. And among them, the marbling is also named also the mean of the intramuscular fat percent, will have crucial roles in the meat quality. This figure shows the meat with good value with many marblings. When we look at it under the microscope, we can see many. Many intermuscular fat among the between the muscle fibers. When the intermuscular fat less than 1%, it will impair the taste of meat. But when the intramuscular fat greater than 2%, it will be with good meat quality. And it's reported that both muscular cells and adipocyte cells are derived from the same mesenteric stem cells. And why they differentiated into different cell types, it is regulated by the development of in different cytokines. The major of MSCs developed into myogenic cells, but a small portion of these cells differentiated into adipocytes, which are the basis of the intramuscular fat accumulation that produce marbling. And so, adipogenesis play an important role in the intramuscular fat development, and the process of adipogenesis is regulated by nutritional, genetic, and environment factors, and among of them, because I am from the animal nutrition and feed science department, so our focus on the how the nutrients modulate the process of adipogenesis. However, the genetic factors play the fundamental roles during the course. So we want to find some cell models to reveal the potential genetic and nutritional factors. This figure shows genetic and nutritional and other factors influence the cell signal pathway, and in turn influence the progenitor cells differentiated into different cell types, including adipocytes and muscle cells, as well as fibroblasts. The process of adipogenesis can be divided into four phases. Four phases. Every phase was marked as some important genes. Among of them, PPR alpha and CPPR alpha will play master role during the process of adipogenesis. And of course, this slide shows the signal pathway we have knowing can modulate the process of adipogenesis. However, we are still unknowing how the process controlled in the intramuscular fat formulation. So we want to find some cell models to reveal the potential mechanisms. There are two cell lines. The first is 33L1 fibroblast, which we have selected as the model of the adipogenesis in adipose tissue because it can be induced adipogenesis and lipogenesis in some biochemical or other factors. And the other cell type is C2C12 myeloblast cells. This cell, in some condition, under some condition, these cells can be induced into muscle cells and with some lipid home position. So we select C2C12 as adipogenesis as a model for adipogenesis in myeloblasts. So based on the two cell lines and some factors, we have developed a screening system to find the novel genes involved in the adipogenesis process, and as well as the how to modulate the process. This is the basic features for our screening system. We select MDI region. Oh, I'm sorry. We selected MDI region as the positive control. It induces 33L1 pre-adipocyte induction, and we select beating. A famous nutrient has been reported and inhibit the it's reported the beating and inhibit the adipogenesis in pre-adipocytes. So we select the beating as the positive control for nutrients, which can induce the adipogenesis in adipose tissue. On the other hand, we select another gene, then F205, which have been reported can inhibit the adipogenesis in pre-adipocytes. So, which served as the novel genes and involved in the lipid deposition in adipose tissue. On the other hand, except for the decreased lipid deposition in adipose tissue, we want to increase lipid deposition in muscle tissue. So we also select the C2C12 myoblast as the cell model for the as the muscle cells model. We select host serum as a positive control to induce the myoblast adipogenesis, and beating is also reported as the positive control for nutrients, which can induce myoblast adipogenesis. As for novel gene screening system, we also select the F205 as a positive control to induce myoblast adipogenesis. Based on our screening system, we have screening more than 500 genes which have no functional literatures. Among them, we have selected two novel genes, which named CREBL2 and LI2. Both of these two genes first cloned in our lab, were suggested to as genes involved in the formation of meat quality. Because we just add the information from our screening system, so we want to perform further functional assays to confirm their function and to reveal the potential mechanism. CREBL2, the full name is CMP response element binding protein LI2. This gene located in chromosome 6, and it have 5 exons. However, the pattern of the CREBL2 was confirmed by the first 4 exons, and it's a novel B-zip1 pattern. which means it's located into the nucleus. And at first, we want to affirm its function in adipogenesis. So we have determined its expression during the adipogenesis induction process. And from the result, we can show both the mRNA level and the protein level CREBL2 also increased during the 8 days induced process. So we conclude that CREBL2 is upregulated during 33L1 pre-adipocyte differentiation. And based on bioinformatics analysis, we found CREBL2 and CREB gene maybe have a potential interaction. So we do the halogenization assay to confirm the interaction. From the results, we can say either overexpression of CREB and CREBL2 or dangerous CREB and CREBL2 also located into the nucleus, so they have localized. So CREBL2 localized with CREB in 33L1 pre-adipocytes, which suggests they have interaction. Furthermore, we perform immunoprecipitation assays to confirm the interaction. From the result of both expression and physiological condition, the result also showed CREBL2 immunoprecipitated with CREB proteins in 33L1 pre-adipocytes, which means both of the two proteins can interact each other in overexpressed and physiological conditions. Furthermore, we performed the red O stone assays to confirm the function of CREBL2 in the pre-adipocyte differentiation. From the result, we can say similar to the positive control. Numerous fat droplets can be observed in the CREBL2 overexpressed cells, whereas few lipid droplets appeared in the cells with empty vectors. So from the result, we can conclude that overexpressed CREBL2 protein can induce pre-adipocyte differentiation. And to reveal the potential mechanism, we detect the two master genes, PPR alpha and CPP alpha, during the induction process. And the results showed that overexpressed CREBL2 protein can induce the cell's differentiation consistent with the increase of PPR alpha and CPP alpha, both in mRNA level and in protein level. And it's reported after the cells, after 3G3L1 cells, the induced adipogenesis. It also needs a process of lipogenesis to deposit the lipid droplets. So we also use two assays to determine whether CREB L2 OE expression and induced adipocyte lipogenesis. The first assay is the glucose incorporation rate assay, and the second is 2-deoxy glucose uptake assay. From the result, we can say only express CREBL2 pattern and dramatically increased glucose uptake with higher GLUT1 and GLUT4 pattern expression, which confirm that all express CREB pattern and induce adipocyte lipogenesis. Except for the all expression of the gene's function, we also use short MI interference sequence assays to confirm the function. At first, we designed two kinds of efficient ion interference sequence target CREBL2 for the knockdown of this gene. From the result, we can say both of the two RNA interference sequence can effect knock down the gene's expression, both in mRNA level and protein level. And use the SNRI as a knockdown tool. We detect the effect of knockdown of endogenous CREBL2, how it will function on the process of adipogenesis. From the result, we can say many fat drop assays can be observed in the cells transferred non-silenced RNA when induced MDI region. But if we knock down of the endogenous CREB pattern, both 1 and 2, it can significantly inhibit the adipogenesis process, which suggests that CREBL2 may be ratio during the pre-adipocyte adipogenesis. And we also detect the expression of PPR alpha and CABP alpha to reflect the potential mechanism. From the result, we also say when knockdown of the CREBL2, it will suppress cells differentiation consistent with the decrease of PPR alpha and CABP alpha expression, both in mRNA and protein level. Furthermore, we detect the effect of downregulation of CREBL2 and the lipogenesis process. From the result, we also say both glucose incorporation assay and 2-deoxy glucose uptake assay. The result shows when knockdown of CREBL2 genes, it will suppress adipocytes uptake, glucose uptake, which confirms that downregulating CREBL2 will suppress the adipose lipogenesis, accompanied with downregulated with GLUT1 and GLUT4 genes. And the above result result showed the effect of CREBL2's function and the cell and the adipogenesis. However, we want to know how CREB and CREBL2 interact each other. So we have also performed the further assays to reveal their interaction. Our results showed when knockdown of CREBL2 is CREB is unblocked CREBL2 induced adipogenesis from the either from the cell morphology as well as the PPR alpha and the CPP alpha expression level. On contrast, when depletion of CREBL2 also unblock CREB-induced adipogenesis, either in the cell morphology or the expression of PPR alpha and CPP alpha. Both of the results conclude that both of CREB and CREBL2's function are crucial for the other. It's reported that the phosphorylation of CREB are important for its function in adipogenesis. So we also test the the result of CREBL2 and the phosphorylation of CREB. The results showed that endogenous CREBL2 depletion significantly reduced the phosphorylate level of CREB, either the MI level or per ton level. And this data indicate CREBL2 was important to maintain the transcriptional activity of CREB and the phosphorylation of CREB. On the other hand, we also want to test the effect of CREB on CREBL2. The result showed that endogenous CREBL2 depletion also decreased the per ton level of CREB, especially the per ton level of CREBL2. Phosphorylation of CREB. So the result suggests that the interaction between CREBL2 and CREB might be important to maintain their per ton stability. Taken together, our result showed that CREBL2 is upregulated during the pre-adipocyte differentiation. Certainly, during both overexpression and knockdown condition and physiological condition, CREBL2 interact and was entirely localized with CREB, and both and the overexpression of CREBL2 was sufficient to promote adipogenesis, consistent with PPR alpha and CPP alpha, and increase the lipogenesis accompanied with increased LUT1 and LUT4. When knockdown of CREBL2 genes suppress adipogenesis conversion and lipogenesis. Additionally, depletion CREB could completely block the effect of expressed CREBL2. And LS CREB knockdown decrease CREBL2 per ton level and vice versa. This figure shows the predict mechanism of how CREBL2 and interact with CREB and related to the upstream modulate sequence of the target genes, such as PPR alpha, PPR alpha, CPP alpha, as well as the effectors such as LUT1, LUT4 to influence the pre-adipocyte differentiation and lipogenesis. Of course, the interaction between CREB and CREBL2 promote their protein stability as well as phosphorylation. Based on our result, we conclude that CREBL2 play a critical role in adipogenesis and lipogenesis, where the interaction with CREB. As for the another gene, LI2, the full name is LOC66273S-form2. This gene located in chromosome 7, and it have 5 exons. However, the last 3 exons formed the gene's protein. And the location assay showed that different cytoplasm distribution of LI2 suggests that this gene exert its functions in cytoplasm. And LI2 expression also contributes to the increase during the 8 days differentiation period, both in mRNA level and the protein level. During the 8 days period, we detect the effect of LI2 on the cell proliferation and the cell cycle. The results showed that all expression of L2 in 33L1 pre-adipocyte will result in a significantly lower rate of proliferation compared with the control, which may be induce the cell differentiation. So we determine the effect of LI2 expression on the pre-adipocyte differentiation process. From the result, we also show we also can see numerous fat droplets can be observed in LI2O expressed cells. So we conclude that our expression of the LI2 protein and also promote pre-adipocyte differentiation. We also want to know how many genes involved in the process. Based on the result of Western blot, we can say during the process of LI2 induced process, the master gene PPR alpha and CPP alpha also increase, and the other two gene, two adipogenesis suppressors, PREF1 and JDD153, will be decreased during the process. We also designed to short interference sequence to target knock down the LI2 gene expression. From the result of MI and Western blot, we also see short interference or sequence for target LI2 and effectively knock down the endogenous LI2 expression. And when we use the effective interference sequence in the process of pre-adipocyte differentiation. From the result, we also can see the only few lipid drops can be seen in the cells when compared with the control. So we include LI2 protein knockdown and suppress pre-adipocyte differentiation. And we also determine the the expression of important important proteins involved in the process. From the result, we also find the PPR alpha and the CPPR alpha were decreased when transferred with the interference consequence target LI2 genes, whereas the PREF1 and the JDD153 were decreased, consistent with the FAS gene and the alpha FABP1 genes. And of course, because the LI2 genes show a function to inhibit the cell proliferation, so we also determined why it have such functions. We we test the expression of SPH3 as well as the CREB and the phosphorylation of CREB. The results showed that when knockdown of LI2, it effectively increased the expression of SPH3, whereas decreased CREB, especially the phosphorylation of CREB. So this result suggesting that LI2 will be necessary when adipogenesis process. Taken together, the LI2 genes also is upregulated during adipogenesis, and the LI2 was sufficient to drive pre-adipocyte differentiation, consistent with the PPR alpha, consistent with increased PPR alpha and CPPR alpha. The potential mechanism may be where modulation of the phosphorylation level of transcriptional activity of CREB. And our expression LI2 protein will inhibit pre-adipocyte growth, whereas knockdown of the protein will result in pre-adipocyte apoptosis, where SPH3 activation during adipogenesis. This slide shows the predict mechanism of how LI2 genes exert its function on the adipogenesis and the cell variability and growth. From the figure, we can see LI2 may perform its function from two pathways. The first is the promote CREB phosphorylation and to accompanying to the upstream modulation sequence of the target gene, such as CPP alpha, PPR alpha, and JDD153. The other pathway is maybe it block the activity of PIF1 and therefore to induce to inhibit adipogenesis and promote differentiation. In conclusion, LI2 functions to switch pro-adipocytes from proliferation to differentiation, and also maintain the pre-adipocyte viability during adipogenesis. Based on our results, the International Genome Naming Committee named LI2 a new name, AAMDC. It means adipogenesis associated MTH938 domain containing protein. Both of this work have received many helps from many professors and my students. Many thanks to Professor Guoyao Wu and Professor Li, Professor Jing Hao, and Professor Zhou. And this research is supported by several projects. That's all. No more. Thank you.
Are there any questions? Maybe I can start. So, two obvious questions. Have you ever co-expressed both of these proteins together?
Yeah.
What do they have an additive effect? Do they synergize? Because you presented them separately. You overexpressed and knocked them down separately.
To transfer both of these genes to find the interaction of these two genes. Yes, maybe we can try in the future. We just from the subcellular location assay find CREBL2 located in the nucleus and LI2 located in the cytoplasm. So from the space, they cannot match each other. So we haven't do the experiment before both of the two genes function.
I see. But you did show that the LI2 can enhance the phosphorylation of CREB, which interacts with your CREBL2, right?
Yeah.
So they do have a relationship or potential indirect interaction.
Yes. Both of them have important target in CREB. So maybe we can try to find the interaction from this rule. Please.
Great study. Curious. I know you're interested in the meat quality, but I'm curious in the physiological relevance.
Physiological, yeah.
Regarding, for example, like from the population study or in the animal model, we need lean versus obese condition. You see the gene upregulated or is there any difference? That's the first question. And another question, talk about the interaction with people gamma, particularly when the people gamma agonist, most popular drug for insulin sensitizer or effects. So do you see any effects in response to human insulin treatment or using insulin treatment?
Oh, I'm sorry.
Okay, so back to
the
first question. So like a lean versus obese condition. So in adipose the gene, do you see any difference?
Both of the gene have the role and the lean meat.
Yeah, another thing. You're interested in the meat quality. I'm asking like a human, for example, like a
for human,
person versus obese person, or animal model versus lean mice versus obese mice. In adipose tissue, do you see difference in gene expression?
We also do the experiment in use the mouse model to find some nutrients, the role of some nutrients and genes and the fat and the above and thin person to reveal its mechanism. But we found because in the mouse model, we need to knock down the genes expression. It will have the knockout mouse will be the other model to study this gene's function. For now, we haven't at the model. Yeah.
So in your cells, have you ever tried to treat the cells with the purism or rosiglitazone? I mean, the people gamma agonist. Do you see any effects? So I know you probably upstream of the people gamma, but we have people gamma agonist, the drug widely used for diabetes treatment.
Yeah.
Do you have any data on gene expression in response to people gamma agonist treatment?
I haven't do such experiment. Maybe yes. The research and the meat quality are similar to the research on obesity and L1 and as well as sometimes diabetes. I think it's also have the it's also a good model for the diabetes and obesity.